In 2007, a grave was discovered near Ekaterinburg, Russia. It contained 44 bone fragments and teeth that had showed evidence of having been damaged by fire and possibly sulfuric acid. This grave sat 70 meters from the grave that contained 9 individuals, identified as the last Tsar of Russia, his wife, 3 of their 5 children, and the 4 servants who had died with them in 1918. When the first grave was excavated, some used the fact that 2 of Nicholas and Alexandra's children weren't found in the same grave to argue that these could not be the remains of the Romanovs. As we've discussed before, that argument is based on the assumption that they all shared the same grave following the execution. There was also a lot of disagreement over who was missing. It was clear that Tsarevich Alexei hadn't been buried in this first grave. All of the skeletons in the first grave belonged to adults. Their bones had completed fusion and development. We would refer to them as having reached skeletal maturity. Alexei was only 13 when he was executed, so his bones would look very different from those of an adult. Skeletal analysis of the remains found in the second grave and testing the Amelogenin gene showed that it contained 2 individuals, one of whom was a male between the ages of 10 and 14. It's the identity of the missing daughter of Nicholas and Alexandra that caused the disagreements. The couple had 4 daughters, the Grand Duchesses Olga, Tatiana, Maria, and Anastasia. The forensic anthropologists from the United States who examined the remains from the first grave thought Anastasia was missing, but the Russians thought Maria was missing. The other individual in the second grave was a female between the ages of 18 and 23. Maria was 19 and Anastasia was 17 at the times of their deaths, but what about a genetic relationship between the occupants of that second grave? The scientists carried out the same types of DNA tests for the second grave's occupants that were done for the first grave: short tandem repeats, or STRs, and mitochondrial DNA. These tests were carried out by 2 independent labs, the Armed Forces DNA Identification Laboratory in Maryland and the Institute for Legal Medicine in Innsbruck, Austria. First, they compared the short tandem repeat patterns of the 2 individuals from the second grave to each other. We share half of our STRs with our biological mother and the other half with our biological father, so we should share some of those chunks of DNA with our siblings, right? These 2 individuals shared a high degree of STRs and alleles, which are different variants of genes, suggesting a close genetic relationship. Their mitochondrial DNA sequences also matched each other, which is what we would expect from siblings who share the same biological mother. This study also involved retesting the individuals from that first grave identified as Nicholas, Alexandra, and 3 of their daughters, and again used descendants of the maternal lineages of Queen Victoria, Alexandra's grandmother, and Empress Maria Feodorovna, Nicholas's mother, as references for the mitochondrial DNA sequences. When compared to living maternal relatives of Maria Feodorovna, the sequence from the skeleton identified as Nicholas matched them again, except for the heteroplasmic mutation at position 16169. They named this sequence the Dagmar mitotype. Nicholas's mother was born Princess Dagmar of Denmark, and it was usual for women marrying into the Romanov family, especially to the Tsar or the heir to the Russian throne, to change their names. They also didn't find anyone with a matching mitotype in a Danish database for mitochondrial DNA, indicating that this sequence is rare. The mitochondrial DNA sequences of the individuals identified as Alexandra and 3 of her daughters matched each other, replicating the results of the original study, and they matched both individuals from the second grave. The 2 individuals from the second grave also had mitochondrial DNA sequences that matched living relatives from Queen Victoria's maternal lineage, which was named the Queen Victoria mitotype. When they compared the Queen Victoria mitotype to databases that included mitochondrial DNA data from over 70,000 individuals, including populations from Russia and East and West Europe, they only found 1 individual who shared the Queen Victoria mitotype. This rarity supports the idea that Alexandra, her 3 daughters, and the 2 individuals from the second grave are maternally related to each other. When they retested the short tandem repeat profiles of the individuals from the first grave and compared them with the occupants of the second grave, they obtained a distinct profile for each person. So, we have 7 distinct individuals, and each of the 3 daughters and the 2 individuals from the second grave shared half of their genetic variants with Nicholas and the other half with Alexandra. This is consistent with parent-child relationships and the biological kinship relationships of immediate family. But we're not done. There's more DNA ground to cover. They also tested the Y chromosome short tandem repeats of Nicholas and the male individual from the second grave. These are repeating chunks of DNA that live on the Y chromosome, so they're passed from father to son, and that makes them useful for tracing paternal lineages. Living relatives were found among the descendants of Tsar Nicholas I. He was the great-grandfather of Tsar Nicholas II, so his male descendants, including Tsarevich Alexei, would share the same Y haplotype. Okay, So what about our YSTR family tree for Tsar Nicholas? I'm going to use a different color here for this. We're getting fancy now. So Nicholas is going to share his Y chromosome STR patterns with his only son, Alexei. And he would share it with George as well. That just wasn't tested. So we need to go up to Nicholas's father, Alexander the Third, his father, Alexander the Second, and then the first Nicholas, and then from that first Nicholas, we have male line descendants who donated their DNA samples for comparison with Nicholas and Alexei. Named among those male line descendants who we got a comparison sample was Andrew. The Y chromosome haplotype of Prince Andrew Andreyevich Romanov, a male line descendant of Tsar Nicholas I and a distant relative of Tsar Nicholas II, matched both the individual identified as Nicholas II and the young male individual from the second grave. You might be thinking that all of this is enough DNA evidence to establish the identity of Nicholas's remains, but wait, there's more. A blood-stained handkerchief wasn't the only artifact from the day Nicholas was attacked by a random sword-wielding dude in Japan. The shirt he had worn that day, now covered in bloodstains, had been preserved in the archives of the Hermitage Museum in St. Petersburg. The scientists took samples from 4 different locations on the fabric and extracted DNA at 2 labs, the Vavilov Institute of General Genetics in Moscow and the University of Massachusetts Medical School. They found multiple different mitochondrial DNA sequences, which does indicate contamination, but one of those sequences matched the skeleton identified as Tsar Nicholas, matched his maternal relatives, and had the cytosine and thymine point heteroplasmy at position 16169 found in both Nicholas and his brother George. The Y chromosome short tandem repeat profile from the shirt's blood samples also matched the skeleton. So far, this is a considerable amount of data validating the results of the original study and supporting the hypothesis that the second grave contained the remains of Tsarevich Alexei and one of his sisters. To further explore whether or not the young male individual in the second grave was Alexei, the scientists look for genetic evidence of the blood disorder the last heir to Russia's throne carried during his short life, hemophilia. After he was born in 1904, it became very clear that Alexei had hemophilia. When his umbilical cord was cut, his navel, his belly button, wouldn't stop bleeding. His blood wouldn't clot. When a person has hemophilia, they don't have enough proteins called clotting factors that allow their blood to clot as part of the healing process after an injury. A person with hemophilia might bruise easily, or a small cut might result in a lot of bleeding. There are 3 types of hemophilia, called A, B, and C, and each type is low in a different clotting factor. Hemophilia A and B are the most common types. Hemophilia can be acquired if a person's autoantibodies, which are proteins made by the immune system that attack healthy tissue, start attacking one of those clotting factors, clotting factor 8. However, hemophilia is a genetic disorder, so it can be inherited or be the result of a spontaneous gene mutation early in fetal development. At the time of Alexei's birth, it was recognized that it ran in families. Specifically, it ran in the family of Britain's Queen Victoria and had been passed to her descendants. It's often referred to as the royal disease, but it did have tragic consequences and no treatment existed at the time. So how does inheriting hemophilia work? Okay, so we have these 3 types of hemophilia, A, B, and C, and each is low in a different clotting factor. Hemophilia A is low in factor 8, hemophilia B is low in factor 9, and hemophilia C is low in factor 11. Each of these factors gets their own gene that lives on the X chromosome, and genes come in different variants called alleles. So there's a variant of the factor 8, 9, and factor 11 genes that produces enough of their respective clotting factor, and a variant, or allele, of each of these genes that doesn't produce enough of that clotting factor. The X chromosome, where these genes live, is one of the chromosomes that plays a role in whether a person is biologically female, with 2 X chromosomes, or biologically male, with 1 X and 1 Y, although there are other combinations of these chromosomes. The first X comes from a person's biological mother. A biological father will contribute either a second X chromosome or a Y. The Y chromosome doesn't carry any genes for these clotting factors, so the combination of an X chromosome with one of those gene variants that causes hemophilia A, B, or C, and a Y chromosome that doesn't have genes involved in the production of any clotting factors will result in a male individual with hemophilia. There is a lot more that could be said about hemophilia, and it deserves its own video, but that is what is relevant for Alexei. So, how can we test human remains for hemophilia? The key is knowing which genes and which variants of those genes cause hemophilia. The scientists used multiplex target amplification with polymerase chain reaction, PCR, to target the F8 and F9 genes, the genes associated with the most common types of hemophilia, A and B. Polymerase chain reaction is a method for making copies of DNA, which makes it easier to study, but you can also use it to test for the presence or absence of genes in a sample. Combining that with massively parallel sequencing, which is the same thing as whole genome sequencing, and I don't know why we have multiple names for the same thing, the scientists looked at the sequences of those 2 genes in Alexandra, who would have passed these genes to her children, and the remains thought to belong to Alexei and his sisters. They found a mutation in the F9 gene of Alexandra, one of her daughters, the skeleton presumed to be Anastasia based on the skeletal age estimation, and the young male individual from the second grave. This mutation made the F9 gene less functional, resulting in hemophilia B, which for some reason is also called Christmas disease. I don't understand why, so I will need to go and look this up later. This supports the hypothesis that these remains belong to Tsarevich Alexei, because we know he lived with severe hemophilia and shared that gene mutation with his mother and sister, whom we would refer to as carriers of hemophilia. All of this genetic data from the reevaluation of the remains from the first grave and comparison with the DNA tests of the 2 individuals in the second grave led the teams of scientists to this conclusion. All of the Romanovs had been found and identified. Tsarevich Alexei and his sister, they identified her as Grand Duchess Maria based on the age estimation of the bones, fitting with her known age, could be reunited with the rest of their family and their ancestors in the Cathedral of Saints Peter and Paul in St. Petersburg. This did not happen. This is not the end of the Romanov DNA saga, but in part 5 of this series, we'll see DNA studies that push the boundaries of molecular anthropology. Thank you for watching.